everybody. Again, the new Xylella Files seminar. We're very excited to have Lindsey Burbank. I think it's the first time that she's presenting in this seminar series. Some people from your lab presented before, but this is the first time, so we're very excited to learn more about your work. And Lindsey, so she got a PhD in University of California Riverside. And the bachelor's degree was in the pathobiology, University of Connecticut. And then after her work as a PhD, she joined the research as a research plant pathologist at the USDA, United States Department of Agriculture, the ARS service in Parlier in California. That, as you probably guys know, the climate in the country is changing daily. So we all hoping that things will stay strong for the USDA and for everybody here. And Lindsey today is going to tell us about the screening for Xylella fastidiosa genes important in plant infection with transposon mutagenesis sequencing. So thank you, Lindsey, again for giving this talk, and go ahead.
Oh,
All right.
From the beginning? Yeah.
All right.
Okay.
There you go.
Good. Thank you. So I'm going to talk today about transposon mutagenesis sequencing in Xylella to identify some virulence genes. And the reason I chose this project specifically is even though it's a little bit in the preliminary stages, there's a lot that can be improved, I think, on these methods for Xylella. I thought it would be maybe useful to other people who might be thinking about similar approaches, or people are using similar approaches in Xylella, to just kind of walk through what I've tried, what's worked, what hasn't worked as well, to help inform the research community. And hopefully we can all build off of this and maybe get some new insights using these methods. So first, I just want to thank some of my collaborators from UC Riverside who helped on some of the components of this project. That was very helpful to me. A lot of this was done in the time between now and 2020. So there was a lot of delays in many things, and they were very helpful. Also, some of my staff here at the USDA who did a lot of work on the plant sampling and things like that. So I don't really need to introduce Xylella to this group, but I just want to very briefly mention how much we do actually know about Xylella and how much research has been done on this pathogen. And so we do understand a lot about how this pathogen is transmitted between plants, how it colonizes the xylem tissue, how it spreads through plants, and how plants respond to it, and what causes some of the symptoms that we see. And a lot of this research is based on several decades of biology work. A lot of it was using gene deletions in the bacteria to understand which genes and which pathways are important for it to be able to cause the disease process. And so there's a lot of work that's been done, and we do actually have a lot of answers about many things related to Xylella. But we also don't have an answer to everything. And these are 10 challenges about Xylella from a review published by the hosts of the seminar series. And I just wanted to point out a couple because I think they are really important. One is the idea of host specificity. So we know that Xylella can infect hundreds of different plant species, but we don't really see severe disease in most cases of Xylella infecting plants. We see that in certain strains of Xylella in certain hosts, but we don't see that in everything. And we still don't entirely understand what makes this a very severe pathogen in some cases, but not in many others. We also have done most of this research on only a few plant species. These are mainly crops. So grapevines, citrus, and olives have been big topics of research for Xylella. But there's also, again, many other plants that can be infected with this pathogen, and we still know very little about many of those systems. And a lot of this is due to technical challenges in working with some strains compared to others. The Pierce's disease strains tend to be easier to work with in many ways. But I do think it's important that we also get some insight from some of these other systems to better understand how maybe the next new disease epidemics will show up. And so what are we missing? I mean, we're missing a lot of things. And today, in the days of genomics and all these other omic technologies, we can start to get at these questions using some larger scale approaches. And many of you have worked on genomic techniques, looking at a wide variety of Xylella strains and sequencing genomes, and looking at the evolution and the adaptation of Xylella in different hosts and things like that. And this has really gotten some interesting insights into Xylella and into some of these questions about how it's evolved and why it causes disease in some cases and not in others. But I also wanted to try to approach this from a little bit of a different angle, and to start with the function and then go back to the genome and see what shows up. But I also wanted to do this using, again, these larger scale approaches that can help get to some of the insights that we can't get at individually looking at genes one at a time. And so this is where a technique called transposon mutagenesis sequencing, or TNSeq for short, comes in. And basically the idea here is that you can make random mutations in all of the genes in the genome, or as many genes as you possibly can in the genome. And then you put that pool of mutants through some selective process, whether this is a selective growth condition or it's through an infection in the plant. And then on the other side, you can sequence all of the mutants that come out on the other side, and you can see which ones have survived, which ones have not survived. And this can give you a lot of insight about which genes are essential under that specific condition. And people have been using TNSeq for a long time, probably around since 2009, 2010 in bacterial research. It's been very useful in a lot of species and a lot of different cases. A lot of this is looking at genes that are essential under different growth conditions, but it can also be used to look at which genes are essential or important for survival and disease in a host. This has been done a lot in animal hosts with various animal pathogens, but it's been largely underutilized, I think, in looking at plant hosts. And so more recently, you can see there's a few papers that have been published on using TNSeq in plant hosts. Especially in the last few years, there's been a lot on things like Ralstonia solanacearum and some other pathogens in plants. But in general, I think there's a chance for us to use this in a lot of ways now that sequencing is so much cheaper than it used to be. And so my inspiration to try this in Xylella came from this paper, which is not about Xylella, but it's about Pathobiology stewartii, which is a different xylem infecting pathogen. It causes Stewart's wilt in corn. And it caught my attention because I did my PhD working on Pathobiology stewartii in corn. And this is a paper from Ann Stevens' group. It was published in 2018. That tells you how long I've been thinking and trying to implement this project. But I saw this and I said, okay, if they can use this to look at bacteria growing in the xylem in this system, why can't I do this also in Xylella? So the idea, again, is to start with a random pool of mutants in Xylella, and then to inoculate these mutants into plants altogether. And so in this case, I use grapevine and almond as two different host plants to compare, and then also an in vitro media growth condition for kind of a control condition. Then after about 10 weeks of the infection process, we re-isolated all of the bacteria from the plants to see which mutants had survived and which mutants had not. And then we sequenced these to compare what genes are important in the plants. So just to go back to the beginning, I do want to mention a couple of choices that are important for these experiments because they do have downstream implications for what the results mean and how the results come out. So the first thing was the choice of the strain of Xylella to use. We know that there's many different strains that can infect different hosts, and many of them behave a little bit differently. In this case, I chose Xylella fastidiosa, subspecies fastidiosa. This is a sequence type 1 strain called M23, and it was initially isolated from almonds in California. It causes almond leaf scorch, but it also causes Pierce's disease in grapevines. And so it can cause disease in these two different hosts that I wanted to compare. And then the other choice is the choice of transposon that's used to make the mutants. And this one might be familiar to many of you who have done mutagenesis work in Xylella. What I used is the EZTN5 transposon, which is a commercially available vector. And this transforms very highly efficiently in Xylella. And so it was good to use that as an easy starting point. Also, I need to mention that these are the host plants we used. They're both highly susceptible varieties. This was grapevines for Chardonnay. For grapevines, this was Chardonnay. And then for almond, this was a cultivar called Y113-20. These are highly susceptible varieties. And I chose to start with the most susceptible varieties I could find, mostly because some of the insights you get from this type of experiment involve being able to isolate a lot of bacteria from the plants at the end of the experiment. And so these two cultivars will give you a very high level of bacterial infection. So we made the random mutants in M23. We inoculated them into grapevines and into almonds, and then used growth media as a control. This was done in 3 separate replicates for each treatment. And then afterwards, we re-isolated all of the bacteria and pooled the DNA from each sample for sequencing. So to do the sequencing, the libraries are made using custom primers, which target the sequence of the transposon. And so the idea here is to specifically amplify the region right next to the transposon to identify the insertion site. And so these were sequenced on the Illumina platform to generate a lot of very short reads. And then they're mapped to a reference genome. And the genes where they map to is identified. And so then for each gene, there's assigned an insertion index. The insertion index is just the number of unique insertions or mutants in that gene normalized to the length of the gene, because obviously different genes will have different lengths and therefore a higher likelihood of having more mutants in there. And so from these experiments, if you map the insertion indexes for all of the genes, what you see typically is this bimodal distribution where you have on the right the peak, which corresponds to genes that are or mutants that are present in high frequencies in your sample. And so if the mutant is present in high frequencies, this indicates that those genes are not essential to grow under that condition. The bacteria is replicating quite a lot. And then you also have this other peak, which corresponds on the left, which corresponds to mutations that are missing from your sample. And this suggests that maybe they are essential for survival under that condition, or they're underrepresented because having those mutations creates a fitness defect. And so if we look at what comes out from the different samples and what causes the variability between the different samples, the largest variance comes from comparing the control, which is growth in media, to the growth in the plant. And so there's some variability between samples, especially for grapevine for some reason, not as much in almond, But the biggest variance is comparing samples that came from the plant versus the samples that were just grown in media. And there's a couple other important caveats for this type of experiment. One of the really important ones is the idea of an inoculation bottleneck. And so this is just the fact that when you inoculate plants with a large population of bacteria, only a small portion of those bacteria will actually become established in the plant and lead to your infection. And so this creates the bottleneck effect where only a small portion of your inoculum is actually getting established. And so the mutants that are in that small portion of inoculum can dictate what you get at the other end of your sequencing. And so when I tried to estimate this from Xylella by re-isolating bacteria from the point of inoculation after 24 hours, and based on that estimate, it seems like less than 1% of the initial inoculum of bacteria actually gets established in the plant, which is very low. And this does have some implications for how the results turn out. But it's not that different from, for example, animal models where they have done similar experiments where they will inoculate bacteria into mice. You also get a very severe bottleneck effect. So it does cause some variability in the results and some headaches with the analysis, but there's still some useful insights that you can get out of it. And so because of this variability and the fact that a lot of mutants are going to be missing by chance during the inoculation process, what we chose to do was look at the candidate genes that showed up on this screen, only the ones that showed up in all 3 replicates of a given condition. So the candidate genes that showed up in all 3 replicates in grapevine, for example, were the ones that we determined to be important under that condition. And this really is just going to be the tip of the iceberg. Again, there's a lot of things that are missing, and I'll talk later about some ways that we might be able to expand this and get at some of the other things as well. There's also some important considerations in general about what TNSeq will miss. So for example, secreted enzymes might be really important for growth of the bacteria in the plant, but because we're inoculating an entire population of bacteria, mutants that don't secrete those enzymes might be able to still use the enzymes that are secreted by the other cells. And so those types of mutants and those types of pathways typically don't show up on a TNSeq screen anyway. So there's a lot of things missing, but we can start to get at some insights that might be unusual by looking at these candidate genes. One last point is that this does also generate some false positives because you have polar mutations or genes that are disrupted because an adjacent gene is disrupted. And so that can show up sometimes as a false positive. So this is what our relatively short list of candidate genes turned out to be. These are genes that are important in a given condition versus the other ones. So genes that are important in almond versus grapevine or control, in grapevine versus almond, etc. And the largest group of genes that showed up was hypothetical proteins. So these are things we don't really know what they are, but it's useful because this allows us to maybe target some of these genes that are previously completely unknown. Other things that showed up that do make sense are things like tRNAs, which would obviously have implications just for bacterial growth. And then what I like to see that also a few things that have been previously identified and characterized in disease with Xylella were also identified. So hemagglutinins, for example, have been well characterized for being important in disease in Xylella. Same thing with outer membrane proteins. So some of these things that we already knew about did show up on the screen. And then there was a few other things that I wanted to look at a little bit more because they're not something that had been previously identified and really looked into. And so the 2 things that I chose to investigate further by making targeted deletions in these specific genes and then doing more experiments with them have to do with toxin antitoxin systems and with phage-associated proteins. And I chose these because, again, these are things that hadn't really been investigated, and I wanted to see if the genes that we're picking up on the screen are in fact important for disease. So just some brief background on antitoxin systems, because that's one of the genes that I chose to highlight here. Antitoxin systems are systems of two different genes that you often find in bacteria. There's somewhere between 10 and 20 predicted antitoxin systems in Xylella, depending on which strain you're looking at. And these are often in the chromosome. They can also be on plasmids. But the general premise is that you have these two genes. One of them makes a toxin. This toxin causes growth arrest or other problems in the cell. And then you have an antitoxin, which interacts with the toxin and prevents it from having whatever activity that it has. And typically these two are produced together. And as long as they're always there together, you don't get a lot of detrimental effects on the cell. But if you were to take away the antitoxin, you can get effect on growth. Also, some of these systems are regulated by being in different growth conditions. And so sometimes they have to do with formation of biofilms, or they can be turned on or off under specific growth conditions, and they can have an impact on either the rate of growth or things like formation of biofilms, among other phenotypes. And there's some background for research in antitoxin systems. Some I've done myself, some that other people have done. This is just one example. These are mutants in 6 different antitoxin systems that I made in some previous work. And when you look at the cell aggregation phenotype, which is very characteristic of Xylella growth in culture, some of these TA systems can have an impact on that cell aggregation, which is an important part of biofilm formation. So over here on the right, you have the wild type, and then you've got a few mutants that are not any different than the wild type, and then some that that aggregate a lot more in culture. And so these systems, even if you take out the entire system and not just the toxin or the antitoxin, they can have an impact both on cell growth and also on things like biofilm formation. This is just another example. This is something I published a few years ago, where if you delete the DI and J RELE antitoxin system, you get faster growth of the bacteria in the plant, and you get more serious disease phenotypes. So this mutant compared to the wild type, you have higher bacterial populations in the plant. It seems to grow faster and spread faster. You also get much more severe disease that shows up more quickly. So in this case, it seems like having the antitoxin system in the genome slows down the growth in the plant a little bit. And that's an interesting thing to be looking at, I think, as far as what exactly these are doing, how they're regulating growth, and how they're regulating disease. And so back to the results of the TN-seq screen and the 3 genes that I chose for further validation. These were all genes that showed up as important in both of the plant conditions, so including almond and grapevine. One was a phage-associated protein, one was the toxin component of a antitoxin system, and then I chose a hypothetical protein just to look at what was going on with that. And so first, we just made deletion mutants, we made complemented strains, and we looked at how they grow in culture. There doesn't really seem to be much of a difference between the mutants and the wild type looking at growth in culture. They seem to grow just fine. There also wasn't really any obvious difference in the aggregation and the biofilm phenotypes of these when they're growing in culture. So that was just kind of a starting point. But again, these were chosen because they were candidates for being important in the plant conditions. So we also did inoculation experiments with all of these. We inoculated them into both grapevines and almond. I'm only showing the grapevine data here, but the results were similar in that they can still infect the plant, they can still cause some disease, but it's not quite as much, not quite as severe. And so you've got on the right the wild type and all of the complemented strains, and then the 3 deletion mutants. And this was interesting because it really shows, I think, that the TNC screen is picking up things that are not specifically essential in the plant, but probably they have some type of fitness defect, and that's maybe why they were underrepresented in the pool of mutants that we sequenced from the plant. We also looked at bacterial population. So this is using qPCR from petiole samples. They are all able to replicate in the plant. You do get a significant amount of bacteria. This was done at 8 weeks post-inoculation. So it's possible if we sampled sooner, there might have been more of a difference in how quickly they're growing or fitness defects in the plant. One thing that was interesting, but I'm not sure how biologically significant, is that 2 of the mutants had much higher variation sample to sample in the bacterial population. Most of this is driven by just having more samples that were undetectable. So there was a little bit more variability there. Not sure if that is a real biological phenomenon or not. But again, maybe if we sample earlier in the infection process, some of these things would be able to stand out more. Just a couple other things I want to point out in terms of what the results mean and how to interpret some of this. Competition is an important part of these experiments. When we do the initial TNC screening, all of the mutants are inoculated together into the plant, and so they're competing with each other. When we do individual inoculations, they're not competing with other strains. And so some of the mutations that maybe have a steeper fitness defect in a competition scenario maybe don't as much when they're inoculated individually. And so this could be one reason why things that show up in the TNC screen are not things that are always completely essential in the plant, but they're things that might have some kind of fitness defect, and that might be more exaggerated in a competition scenario. The other point I wanted to touch on a little bit was the host susceptibility. So again, I chose the most susceptible plant I could find because I wanted to be able to re-isolate a large population of bacteria. It's possible that the results would be a lot different if it was a less susceptible host, and then maybe more pressure from the host response and the immune response to the bacteria. So there's a lot of things that I think could still be answered a lot better in future experiments. And so I'm going to talk a little bit about what I think can be useful to think about in the future. If anyone else is working on similar things, I would love to hear what your results have been and what you've tried. These are just things that I've tried so far and that I'm still working on. So first, I think there's a lot to be done about improving transposon mutagenesis to create these random mutant pools in the first place. I use a commercially available transposon. It's highly efficient, but there's no guarantee that that's available to everybody. It's expensive, and it might not always be a product that is still available. And so I do think it's important to think about what other vectors for mutagenesis we can use that are equally efficient, but maybe more easily available to everybody. Part of that is I know there are vectors available that can do barcoding of transposon insertions. If anyone has one, that is as efficient in Xylella, I would love to hear about it. I'm interested in looking into using some of those too. Also, improve transformation of other strains and other subspecies. So I use a sequence type 1 strain that's on the overall spectrum of Xylella, relatively easy to transform, and so you can get higher efficiency of creating mutations. Many of them are not so easy, and I think in order to get some insights from other strains, we do need to look at if we can improve transformation for those. Improving the experimental design as well. So I talked a lot about the bottleneck effects that you can get when you're inoculating Xylella and how this creates variability in between samples. One of the things that might help with this would be collecting samples from a wider range of plants and doing more pooling of samples. There's always some trade-offs here between how many samples can you sequence versus what information you're getting from it. But I think looking at things like pooling sample strategies might be very helpful. Also, more targeted samples. This experiment was intended to target the entire disease process, but maybe it would be more helpful in cases where you're looking at, for example, movement within the plant. So taking samples from different points up the stem, and specifically looking at those things instead of trying to look at the whole disease process all at once. Also addressing host susceptibility. I mentioned this a little bit. There's a lot of different pathogen host combinations. There's tolerant versus susceptible materials. And there could be a lot of information to be gained, I think, from looking at more tolerant material and what genes in the bacteria are important for surviving in that condition. That is challenging, again, because you have to have a host where there's enough bacteria that you can isolate live bacteria at the end. And so with that, I'm going to end here. This is a little bit shorter seminar than many, but I thought it would hopefully be useful to anyone doing similar work. I have a preprint on this project that's available if anyone is interested. And please do reach out to me if you're doing similar things. I would love to know what other people are trying as well. Thank you.
Thank you, Lindsey. Thank you for the presentation, great presentation. So now we open for questions, of course, so people can raise their hands and talk or writing in the chat, and Lindsey can answer to your questions.